Hi dear students. So today let's discuss about culture methods. Culture methods employed depend on the purpose for which they are intended. In the clinical laboratory, the indications for cultures are mainly to isolate bacteria in pure cultures, to demonstrate their properties, to obtain sufficient growth for the preparation of antigens, and for other tests. It's mainly involved in the development of vaccines and an immunological test. Then for bacteriophage and bacteriocin susceptibility. A bacteriophage is a type of virus that infects bacteria. And the bacteriocins are proteinaceous or peptidic toxins that is produced by bacteria to inhibit the growth of similar or closely related bacterial strains. And the next is to determine the sensitivity to antibiotics, to estimate the viable counts, and to maintain stock cultures. Culture methods include streak culture, lone culture, stroke culture, stab culture, pour plate method, liquid culture, anaerobic culture methods. Let's see this one by one. The first one is streak culture. It is used for the isolation of bacteria in pure culture from clinical specimens. Platinum wire or nichrome wire is used for this. One loopful of the specimen is transferred onto the surface of a well-dried plate, and it is spread over a small area at the periphery. The inoculum is then distributed thinly over the plate by streaking it with a loop in a series of parallel lines in different segments of the plate. On incubation, separated colonies are obtained over the last series of streaks. You can see how a streak plate is done here. You have to take a petri plate and then a loop. It can be a nichrome resistance wire, and one loopful of the specimen is transferred onto the surface of a dried plate, and it is distributed thinly over the plate by streaking it with loop in a series of parallel lines. Then different segments of the plate can be streaked similarly, and in between the loop should be flamed and cooled between the different sets of streaks. Like after completing the streaking here, we have to flame and cool the loop, and then again streak it again, then flame it and cool, then again streak it. So after that, on incubation, growth may be confluent at the site of original inoculation, but it becomes progressively thinner and well separated colonies are obtained over the final series of streaks. This is the petri plate after incubation. So you can see here the confluent growth, and it becomes progressively thinner, and at the end you can see separated colonies towards the end. Once again, let's see how the streaking is done. You can see the first plate here, and then after streaking these parallel lines, we have to flame and cool the loop. Again the next set of parallel lines is being streaked, then again flame the loop and cool it, then the next set, then again flame and cool, then the next set, you can see it in green, and finally after incubation you can see the growth of microorganisms in this pattern, and at the end you can see isolated colonies like this. Lawn culture is also termed as carpet culture. It provides a uniform surface growth of the bacterium and is useful for bacteriophage typing and in antibiotic sensitivity testing. It may also be employed when a large amount of growth is required on solid media. for example, in the preparation of bacterial antigens and in vaccines. Lawn cultures are prepared by flooding the surface of the plate with a liquid suspension of the bacterium. Alternatively, we can also use a swab soaked in the bacterial culture or suspension for lawn culture. We can also say it as a swab culture. This is an example of lawn culture after incubation and antibiotic sensitivity testing. Initially, you can see that here there will be full of the growth, and this zone of inhibition is seen. It is due to the activity of the antibiotic that has been added to it. So this is mainly used for antibiotic sensitivity testing and for the bacteriophage typing. Next is a stroke culture. It is done in tubes containing agar slopes, and it is used for the pure growth of bacteria and also for biochemical testing and other diagnostic tests such as agglutination, slide agglutination test and all. These are all immunological tests performed. In this technique, you can just make a stroke over the agar slope with the loop. Next is the star culture. It is performed by a straight wire charged with culture material by puncturing deep inside the agar. It is used to demonstrate gelatin liquefaction, to identify the oxygen requirement of bacteria, to maintain stock culture for the preservation of bacteria. Next method is the pour plate culture method. Here, the agar medium is melted and cooled to 45 degrees Celsius. 1 ml of the inoculum is added to the molten agar and it is mixed well and poured to a sterile petri dish. And it is allowed to set, and we have to incubate at 37 degrees Celsius. Colonies will be distributed throughout the depth of the medium, and it is used to give an estimate of the viable bacterial count in a suspension or a sample. And also for the quantitative urine cultures also we use the pour plate method. Let's see how the pour plate method is performed. Inoculate the empty plate with 1 ml of the sample. After that, melted neutron agar is added to the petri plate. Slightly swirl to mix the inoculum and the neutron agar, and we can keep it aside for setting. After incubation, we can see that the colonies grow in and on solidify the medium. This is the method for the pour plate method. Just like that, there is also another method called the spread plate method. Let's see how the spread plate method is performed. For this, we have to take a petri plate with a solidified agar in it. And from the sample, that is from the bacterial dilution, take 0.1 ml. But in pour plate, we took 1 ml. But in spread plate method, we are taking 0.1 ml of the sample. Then pour it over the solidified agar medium in the petri plate. Then with the help of a L rod, spread the inoculum over surface evenly. Then keep it aside for setting. After incubation, you can see the colony grows only on surface of the medium. So let's once again see the main difference between the pour plate method and the spread plate method. Both methods are used for the isolation of microorganisms. In pour plate method, 1 ml of the sample is used, whereas in spread plate method, 0.1 ml of the sample is used. Here the 1 ml of sample is poured to the empty petri plate, and here 0.1 ml of the sample is poured over the solidified agar medium. And later, after incubation, we can see that in pour plate method, the colonies grow in and on solidified medium. But whereas in spread plate method, the colonies grow only on the surface of the medium. The next method is liquid culture. Liquid cultures are done in tubes, bottles or flasks and may be inoculated by touching with a charged loop or by adding the inoculum with pipettes or syringes. Large inocular can be employed in liquid cultures and hence this method is adopted for blood culture, for sterility test, for biochemical examination, and when large yields of the bacteria are required, we use liquid culture. And the yield can be further enhanced by aeration, addition of nutrients and the removal of toxic metabolites. And this technique is mainly employed in continuous culture methods, and that continuous culture method is mainly employed in industrial areas.